Welcome back to the deep dive, where we take a stack of complex source material, in this case, the blueprints for life itself, and extract the critical knowledge you need to be truly well-informed.
Today, we are really getting into it.
We're focusing on biological continuity, or, put simply, how a single cell makes two perfect copies of itself.
Exactly. For all the previous discussions we've had on cell structure and function, the intricate dance of the organelles, the selective power of the membranes, none of it matters if the cell can't replicate faithfully.
Right.
This entire process points back to the second basic pillar of cell theory, biological continuity. If cells didn't replicate with incredible precision, genetic stability would vanish and, well, complex life would immediately halt.
And the mechanism that ensures this continuity is what we are diving into. The cell cycle. This isn't just one event, it's the precise sum total of events that must occur between the completion of one cell division and the start of the next.
And this cycle is governed by two non-negotiable fundamental requirements. Requirement 1. The complete genetic material, the DNA, must replicate perfectly. This is followed by nuclear division, or mitosis.
And requirement 2.
The cytoplasm, the organelles, and all the necessary membranes must arrange and partition themselves properly so that when the cell finally splits, you get two complete functional daughter cells. That's cytokinesis.
Fail either of those, and the consequences are bad.
They range from tissue malfunction to cancer. It's that serious.
So why do eukaryotic cells actually bother to put themselves through this complex, high-stakes process?
Well, the necessity boils down to three primary functions. First, reproduction. For single-celled organisms like yeast, division is how they create new individuals.
They just double.
They just double. They don't have sex. They just make a copy. Second, growth. Think about the scale of a human life. We start as a single fertilized egg,
One cell.
and through countless precise divisions, we end up as an adult composed of over 10 to the 13th cells.
10 to the 13th. That means the machinery we are about to discuss has performed that copying operation flawlessly a quadrillion times, minimum. That number alone is staggering.
It is, and the third reason is perhaps the most continuous and underappreciated. Replacement. Our tissues are constantly under stress, either being worn down by the environment or intentionally degraded.
Like through autophagy.
Right, through processes like autophagy. We must continuously resupply that cell pool just to stay even. If you consider just one cell type, we destroy and replace approximately 200 million red blood cells every single day.
Wow, every day.
It's a massive, non-stop maintenance operation.
That maintenance effort really puts into perspective why this cycle is so critical. Okay, let's unpack this cycle. We're going to focus on the key stages G1, S, G2, and M, and the molecular switches that control them.
Right.
What's truly wild here, and a major theme of this deep dive, is the incredible conservation of the control mechanisms.
That is the core, profound insight we take away from the sources. Whether you look at a simple budding yeast cell, a rapidly dividing starfish egg, or a complex human fibroblast, the basic molecular switches that govern when a cell decides to replicate DNA or start mitosis are surprisingly almost identically conserved.
So our mission today.
Our mission is to walk through these stages, structure, function, and the strict internal and external controls, so you understand how this essential process operates and where it can go wrong.
Let's start by addressing the timing problem. If we look across an adult multicellular organism like a human or a mature plant, the rate of division is highly variable.
It really is.
Some cells are dividing constantly and some never divide at all.
That's right. Neurons in the adult mammalian brain or specific differentiated cortical cells in plants are generally non-dividing. They've exited the cycle entirely.
So their cell cycle time is infinite.
Effectively, yes. They have chosen path number 2, permanent quiescence. Contrast that with the highly proliferative areas, which are the engines of growth and replacement.
Where do we find those engines in plants?
Plant growth is localized in regions called meristems. You find them at the root and shoot apex, responsible for primary growth making the plant taller, or in the vascular cambium, responsible for secondary growth.
Making it wider?
Exactly. These are the plant's equivalent of stem cell niches.
And animals have analogs to these meristems, which are, of course, the stem cells.
Precisely. We see stem cells in the bone marrow constantly generating new blood cells, and the crypts at the base of the intestinal villi rapidly producing epithelial cells to replace the lining that is sloughed off.
This is happening constantly.
This relentless replacement activity means that in a healthy human body, Roughly 25 million cell divisions are occurring simultaneously at any given time.
25 million.
It's an enormous nonstop factory.
So if we look at a homogeneous, rapidly cycling population like these stem cells or cells grown in culture, what is the typical cycle time, or TC?
Typically, TC3 ranges from 15 to 40 hours. This is a very broad average, but the key generalization is critical for understanding development. Embryonic tissues and regenerating adult tissues always have significantly shorter cycle times compared to adult differentiated tissues.
They're built for speed.
They are simply prioritizing speed.
Now, we need to address a profound and common misconception, particularly concerning cancer. The instant assumption is that tumors grow fast because their individual cells divide much faster than normal cells.
That assumption, while intuitive, is often incorrect. Data from human tissues show that tumor cells can frequently have longer cycle times.
Longer?
Yes. The source material cites examples. Normal colon epithelium cycles at roughly 39 hours, while a stomach carcinoma might take 72 hours.
That's almost twice as long for the cancer cell to complete a single division.
It is.
Wait, if the cells themselves are slower, why are tumors characterized by rapid, aggressive growth? This seems completely counterintuitive.
This is where the core insight lies. Tumors grow aggressively for two primary reasons that override the individual cell cycle time. First, a much greater fraction of the cancer cells are actively cycling at any given time.
So more of them are in the race.
They've lost the signaling control that would normally push them into reversible quiescence, what we call G0. Second, and critically important, fewer cancer cells die. In normal tissue, the growth rate is balanced by a massive rate of cell death.
But in cancer?
In cancer, that critical process of programmed cell death is inhibited.
So it's not a speed problem, it's a fraction problem combined with an inhibition of death problem.
Exactly. It's a shift in the balance. A slower dividing cell population, if all of them are cycling and none of them are dying, will always outgrow a faster dividing population where maybe 90% of the cells are quiescent and a high percentage are dying naturally.
This has profound implications for cancer treatment, particularly the older generation of drugs that are often mitotic inhibitors.
Absolutely. Many older anti-cancer drugs work by targeting rapidly dividing cells. They act on the mitotic spindle or interfere with DNA synthesis.
But you just said many tumor cells are not dividing quickly.
Exactly. So these drugs are inherently less specific than desired. They inevitably end up killing the body's most rapidly dividing normal cells, such as stem cells in the bone marrow, the intestinal lining, or hair follicles.
Which explains the side effects.
That lack of specificity is why chemotherapy often leads to severe side effects like immunosuppression, gastrointestinal distress, and hair loss.
Speaking of mitosis, let's define the stages. Mitosis, the period where we can cytologically detect the condensed chromosomes, only occupies a fraction of the total cycle.
A small fraction. We quantify this fraction using the mitotic index, or M.
And M is what exactly?
It's simply the fraction of cells observed to be in mitosis at any single moment.
Right.
If M is 0.1, the cell spends 10% of its total cycle time in mitosis.
And the other 90%.
The overwhelming remaining time, typically 90%, is what we call interphase.
Interphase isn't just downtime, though. It's when all the preparation happens. It's subdivided into three crucial parts: G1, gap 1, S, synthesis, and G2, gap 2.
Right.
So how did researchers figure out where these phases start and stop?
They needed clever methods, and the first, the percent-labeled mitosis technique, is a beautiful example of molecular tracking. You pulse a cell population with a radioactive precursor.
Something like radioactive thymidine.
Typically tritiated thymidine, or 3H-thymidine, for a very short period. Since thymidine is incorporated only into newly synthesized DNA, this radioactively tags cells currently in the S phase.
Okay, so you've tagged the S phase cells, then you follow that pulse with a non-radioactive chase, and you start watching the population over time. What happens right away?
Initially, if you look at the cell population under a microscope, you find labeled interphase nuclei, but no labeled mitotic figures. That is proof positive that DNA synthesis, the S phase, occurs during interphase, not during mitosis itself. As you lengthen the chase time, you are tracking a synchronized wave of tagged cells moving through the cycle. The time it takes for those labeled cells to first hit the M phase, that delay before you start seeing labeled mitoses, tells you the exact length of the G2 phase.
So G2 is the gap between the end of DNA synthesis and the start of visible division.
Correct. As the labeled cell cycle, you chart a wave, which you can see in Figure 12.2. The width of that wave estimates the duration of S phase, and the total cell cycle time, or TC, is simply the interval between successive waves of labeled mitosis.
And G1.
G1 is then calculated by subtraction, filling the remaining time gap. It's a way of measuring time inside the cell using radioisotopes. It's really quite elegant.
The second technique, flow microfluorimetry, provides a quantitative snapshot of the entire population instantly.
Yes, and this method revolutionized cell cycle studies because it's fast and quantitative. It relies on staining cells with a fluorescent dye that binds stoichiometrically, meaning quantitatively, to DNA.
One-to-one binding.
Exactly. You then pass these cells one by one, up to 1,000 cells per second, past a laser beam. The fluorescence emitted is directly proportional to the cell's DNA content.
And the output is a histogram, a frequency distribution. What does that distribution actually show you?
It clearly resolves the cell population into 3 distinct groups. The first and often largest peak is the G1 cells. They have a baseline relative DNA content. Let's normalize it to 1.0.
Okay.
The second peak, further down the scale, consists of G2 and M cells. They have exactly twice the DNA content, or 2.0, because they have already replicated their genome but haven't yet separated.
And what about the space between those two peaks?
The cells scattered between the 1.0 and 2.0 peaks are actively synthesizing DNA. They are in S phase. This allows for an extremely precise, large-scale calculation of the distribution of cells across all phases.
So based on all these sophisticated methods, what's the major conclusion about the duration of the phases?
We generalize that mitosis M is short, maybe 1 to 2 hours. S phase, the massive task of DNA replication, is at least 4 hours. But the critical takeaway, shown clearly by data like the developing rat neural tube in Table 12.3, is that variations in the total cycle time, TAC, are overwhelmingly due to variations in the length of G1.
So G1 is the gatekeeper.
It is the highly variable decision-making phase where the cell pauses until it's ready to commit.
That immediately frames G1 as the central control point, which we'll definitely come back to. But to study these phases in depth, especially biochemically, you need all the cells marching in lockstep.
Synchronization is absolutely crucial.
Why is that?
Imagine trying to isolate a specific enzyme that is only expressed for 15 minutes during late S phase from a mixed population. It would be so diluted and hard to detect.
Right, impossible to find.
So we exploit natural synchrony where it exists, like in the lilianther, the slime mold physarum, or the rapid early cleavage stages of animal embryos where the entire population divides simultaneously.
But for most research, particularly in culture, researchers rely on induced synchrony. How do they force a whole population into the same phase?
There are 2 main induced strategies. The first is selection. For cultured mammalian cells, when a cell enters mitosis, it typically rounds up, decreasing its adhesion to the culture plate.
Why does it do that?
It's partly due to the reorganization of the fibronectin matrix. So a gentle wash can selectively remove these round mitotic cells. When you place them in new medium, they proceed synchronously through the rest of the cycle.
You can just wash them off.
You can. Alternatively, newly divided G1 cells are smaller, so researchers can use density gradient centrifugation to physically separate smaller G1 cells from the larger ones in G2. You're simply physically sorting them by size and density.
Okay, that's selection. The second strategy uses inhibitors chemical manipulation to block the entire population at a specific point.
This is commonly done by using high concentrations of thymidine, which feedback inhibits deoxyribonucleotide formation. This effectively arrests the entire population at the crucial G1S boundary, preventing entry into S phase.
And then when you wash the inhibitor out,
the entire population synchronously enters S phase. The major caveat here is that chemically blocking a process can cause unintended side effects, like aberrant DNA structure, which might influence the results you're trying to study.
We also see environmental controls used for synchronization, which feel a bit more precise and less chemically invasive.
They are powerful in certain specialized organisms. We see synchronization via light-dark cycles for algae like chlorella, or temperature cycles for protozoa like tetrahymena.
There's a great example with diatoms, right?
A particularly illustrative example is the diatom Nitzschia angularis. This organism requires silicic acid to build its rigid cell wall during cytokinesis. So if you starve the culture of silicic acid, as shown in figure 12-4, the cells progress through G1, S, and G2 until they reach cytokinesis, where they arrest as double cells. Re-adding the acid causes a massive synchronized burst of division within minutes.
Wow, it's like a starting gun.
It is. Finally, we have the immense power of mutants. specifically conditional mutants of the cell division cycle, or CDC, which have been pivotal in mapping the cycle's logic.
So how do these help?
These mutants allow us to define the genetic and biochemical pathways. Conditional, temperature-sensitive mutants are especially useful. They function normally at a permissive temperature, but when you shift them to a restrictive or elevated temperature,
The protein messes up.
the abnormal protein denatures and the cell arrests precisely at the gene's execution point.
And studying these specific arrests helps us map the logical flow, which events depend on which other events.
Precisely. If we look at budding yeast, we can compare two mutants. CDC2 fails to complete DNA synthesis and arrests, while CDC13 does complete DNA synthesis but still fails nuclear division, resulting in the same terminal phenotype.
So what does that tell you?
Since the outcome is the same, we deduce that DNA synthesis must logically precede nuclear division. It's a dependent pathway.
I see.
Conversely, if you look at CDC7, where bud emergence fails but DNA synthesis occurs, versus CDC24, where the reverse happens, you can deduce that bud emergence and DNA synthesis are on entirely separate parallel pathways. You can see this logic laid out in Figure 12-6.
So these tools basically let you build a flowchart for the cell cycle.
Exactly. They define the essential sequence, constraints, and timing of the cell cycle.
Let's dedicate some serious time to interphase, starting with the gatekeeper, G1. We established that its duration is the most variable and dictates the overall cycle time. What's happening structurally and biochemically during this waiting and preparation phase?
Well, G1 is all about achieving sufficient mass and synthesizing the necessary components for the massive task of S phase. The cell and nucleus, literally grown nuclear volume, can double in some plant cells.
And it's making key proteins.
Yes. Key macromolecules are synthesized, such as fibronectin, which is necessary for mammalian cells to reattach after mitosis, and crucially, all the enzymes required for DNA synthesis that will be needed late in G1 in preparation for S phase.
But G1 is most critical because it's the phase where the cell has to make a fundamental lifestyle choice.
Exactly. The cell faces three choices after mitosis. 1. Continue the cycle and divide again. 2. Permanently stop dividing, becoming terminally differentiated like a mature neuron or a muscle fiber. These cells will contain the baseline G1 amount of DNA forever.
And the third option.
Or 3. Enter a state of reversible quiescence called G0.
G0 sounds like hitting the molecular pause button. What separates a cell in G0 from a cell that is just taking a very, very long G1?
That's an important distinction. G0 cells are not just delayed G1 cells, they are biochemically distinct. They have unique non-histone protein profiles and different patterns of RNA synthesis.
What's a good example?
Immune system stem cells are classic examples. They reside in G0 until they're stimulated by an antigen, at which point they're rapidly activated to proliferate.
And we have fantastic experimental proof that G0 cells are actively preventing division rather than just passively waiting.
We do, using cell fusion experiments. If you fuse a G0 cell or inject its cytoplasmic extract into a cell that is currently in G1 or early S phase, the recipient G1S cell stops synthesizing DNA.
So there's an inhibitor in there.
It implied that G0 cells contain a specific diffusible inhibitor, likely identified as a glycoprotein, that actively blocks the progression into S phase. They are in a controlled suppressed state.
The journey from G0 back into G1 and then toward S phase is mapped out as a complex checklist requiring external signals, the growth factors.
This sequence, illustrated dramatically by the mouse fibroblast model in Figure 12-7, highlights how many checkpoints are involved before the cell risks replicating its genome. First, to even exit G0 and move into early G1, the cell requires platelet-derived growth factor, or PDGF.
Okay, so PDGF gets things started.
It drives the earliest events: increased membrane transport and changes in chromatin structure. Next, to stimulate the middle G1 events like increasing the number of polyribosomes and key glycolytic enzymes, epidermal growth factor, or EGF, is needed.
So PDGF gets you started, EGF gets you prepared. What's the final signal needed to commit to DNA synthesis?
Latent G1 to synthesize the critical proteins required for DNA replication, like the specific cyclin A we'll discuss later, and thymidine kinase insulin-like growth factor I, IGF-I, is required.
It's a whole cascade.
It's a strictly sequential multi-factor checklist that ensures the cell is properly equipped and ready before committing its resources to replication.
Given all this complexity, there's an alternative, almost provocative view of G1.
Yes, the alternative perspective proposes that G1 isn't strictly necessary or fundamental to the cell cycle machinery itself. Rather, it exists only because eukaryotic cells grow relatively slowly and simply need time to accumulate sufficient division potential.
Time to bulk up.
Basically, yes. They need the time to grow large enough and make enough internal components to support two cells.
And we see evidence for this accelerated cycle in nature.
Absolutely. Think about bacteria like E. coli, which can divide so fast that its cell cycle time is actually shorter than its DNA synthesis time, meaning it's continuously replicating.
And in eukaryotes?
Eukaryotic examples include the rapid early cleavage stages of sea urchin eggs, where the SM cycle can be less than an hour long, skipping G1 entirely. We even have cultured Chinese hamster cell lines that are laboratory adapted to be deficient in virtually all of G1 and G2.
So they're just on an ultra-fast S to M cycle.
Right. G1 is a sign of a cell that is not maximizing speed.
Once G1 has given the green light, we hit S phase, the DNA synthesis stage. We established that the initiation of replication requires a diffusible cytoplasmic factor.
This factor is crucial, and its existence was proven by cell fusion experiments. Fuse an early S-phase cell body, a cytoplast, with the G1 cell, and the G1 nucleus is immediately induced to start replicating.
But more importantly, there's a constraint, right?
More importantly, this factor enforces the crucial constraint. DNA replicates only once per cell cycle. A G2 nucleus, which already contains twice the DNA, cannot be induced to replicate again even in the presence of this factor.
A critical safeguard.
It prevents catastrophic overreplication.
The details of how our massive linear chromosomes replicate were mapped out beautifully using a technique called fiber autoradiography. This is where we see the physical proof of eukaryotic DNA structure in action.
This is a classic experiment. You pulse S-phase cells with radioactive thymidine, chase with non-radioactive thymidine, and then gently isolate and stretch the DNA strands, exposing them to photographic film. You can see the result in figure 12-8.
So the radioactive sections show up as black tracks on the film.
Correct. And the findings led to two non-negotiable conclusions about eukaryotic replication.
Conclusion 1.
Eukaryotic DNA uses multiple replication origins.
Okay, what did they see that proved that?
The film images showed not one long track, but numerous short, tandemly arranged replicating sections, known as replicons, each one growing outward. A typical human chromosome has a DNA molecule about 4 centimeters long. If it replicated from a single origin, even at the measured rate of about 3,000 base pairs per minute, it would take an entire month.
But S-phase only takes a few hours.
Exactly. So having multiple origins initiating simultaneously is absolutely essential.
That's a staggering evolutionary optimization. And these origins aren't activated randomly, are they?
No, the order is non-random in mature tissues. The transcriptionally active open DNA regions known as euchromatin replicate early in S-phase, while the highly packaged heterochromatin and highly repetitive sequences, which are often silenced, replicate late.
And some organisms can even change the number of origins.
Yes, fast-cycling organisms like the frog Xanthopus shorten their S-phase in early embryos to 30 minutes by activating an enormous number of replication origins and forcing them all to replicate simultaneously.
Okay, so that's conclusion 1. What's conclusion 2?
Replication is bidirectional. When you look at the track patterns, the radioactive label appears on both sides of a central, unlabeled point. This shows that the replication fork moves in both directions, away from the origin along the DNA fiber.
A critical discovery. Now, the physical act of replication also raises structural questions about the DNA polymerase machinery. Is the enzyme moving along the DNA, or is the DNA template moving through a fixed enzyme complex?
In prokaryotes, the enzymes appear to be soluble and mobile. But in eukaryotes, the source material favors the theory that the replication enzymes, the polymerase, are fixed to the nuclear scaffold or matrix, and the DNA template passes through them.
Like thread through the eye of a needle.
Exactly, like in the model in figure 12-9. To initiate replication at a new origin further down the line, the DNA template loops around to meet the fixed enzyme complex.
What visual or experimental evidence supports the idea of these fixed replication factories?
Labeling experiments visualized not millions of individual origins, but several hundred distinct replication foci within the nucleus. You can see this in figure 12-10.
And since there are far more origins than foci,
it suggests each focus must represent an aggregate of roughly 20 origins operating together in a fixed spatial complex, a replication factory fixed to the underlying nuclear scaffold.
we must touch on S-phase specific anti-cancer drugs again, as they are specifically designed to cripple this process.
Yes, they primarily inhibit DNA synthesis by interfering with nucleotide pools. Cytarabine, for example, is a modified nucleoside that inhibits DNA polymerase. Fluorosil inhibits thymidylate synthetase, starving the cell of essential DNA building blocks.
But the specificity is still a problem.
As we reiterated, because all rapidly proliferating tissues rely on this S-phase activity, these drugs lack the specificity needed to exclusively target tumors, resulting in widespread side effects on normal tissues.
Finally, we hit G2, the final gap phase, usually short, lasting typically 1 to 4 hours. What's the main job here beyond ensuring S-phase is complete?
G2 is primarily a final preparation and quality control phase before mitosis. It contains a critical DNA damage checkpoint.
So it's a last look over.
Right. If the cell is exposed to DNA-damaging agents like ionizing radiation, G2 will dramatically lengthen. This pause gives the cell crucial time to activate repair mechanisms before committing to the catastrophic separation errors that would occur in M phase with damaged DNA.
And certain anti-cancer agents exploit this G2 checkpoint deliberately.
They do. Agents like cyclophosphamide cross-link adjacent DNA molecules. When this happens, the replicated DNA molecules become physically tethered and cannot properly separate to form distinct chromosomes, forcing the cell to arrest and remain stuck in G2.
The most surprising experimental insight into G2 might be the cell fusion results, showing just how fast the cell can flip the switch to M phase.
It's incredible. Fusing a mitotic cell with a G2 cell nucleus induces premature chromosome condensation in the G2 nucleus within a mere 30 minutes.
30 minutes? Wow.
It suggests that G2 only needs to be that short to prepare, and that diffusible condensation proteins, a mixture of specialized histones and non-histones made during G2, are responsible for initiating the compaction of the DNA into visible chromosomes.
We see that preparation at the molecular level too, right?
Yes. Other essential G2 events include histone H1 phosphorylation, a prerequisite for forming the higher-order chromosome structures, and the synthesis of tubulin, the raw material for constructing the mitotic spindle.
And just like G1, it's not always necessary.
Exactly. Much like G1, the existence of G2 deficient cell lines suggests that its typical preparatory events are not strictly locked into that time frame. They can occur earlier during S phase if the cycle demands speed.
Now we enter the highly choreographed, dramatic visible phase, mitosis, M phase.
Yeah.
This process is astonishingly accurate and fast, lasting just 1 to 2 hours divided into 5 periods.
Right. Prophase is the longest, and anaphase, the moment of separation, is the shortest.
So prophase kicks off with what?
The primary event, chromosome coiling and condensation. This compaction happens in 2 stages, forming a helical structure, followed by further folding onto itself, a process potentially regulated by calcium levels.
And at this point, each chromosome has 2 chromatids.
Crucially, yes. Each shortened chromosome now consists of 2 identical chromatids, held together at the centromere.
The centromere isn't just a simple pinch point for structure. It's a complex, multifunctional piece of machinery.
Absolutely. Drug studies revealed 3 functional domains in this region. The kinetochore domain is the outer layer, serving as the physical attachment site for the spindle fibers. There's a central region, and finally a pairing domain along the inner surface where the 2 sister chromatids are held tightly together.
And the kinetochore is the key for movement.
It specifically contains structural proteins attached to the underlying centromeric DNA sequences. It's the handle.
What about the nuclear structure during prophase? We need to clear the way for the spindle.
The nucleolus disintegrates early on at the nucleolar organizer regions. Most dramatically, the nuclear envelope breaks down at the end of prophase.
How does it happen?
This breakdown is triggered when the lamins A and C proteins that form the nuclear scaffold, the supporting meshwork inside the nucleus, are phosphorylated, causing them to become soluble.
That's the key step for solubilization. But what happens to the membrane itself?
This is a genius piece of molecular recycling. Lamin B, a different type of lamin protein, remains associated with the actual membrane fragments. These membrane pieces, tagged with lamin B, are temporarily stored in the endoplasmic reticulum, the ER.
So lamin B is like a temporary storage tag.
Exactly. It ensures the envelope can be efficiently reassembled in telophase. Though we should note, this breakdown isn't universal. In yeast and many protists, the nuclear envelope remains intact during division.
Prophase is also when the division machinery sets up the plane for division.
Yes. The centrosomes, the cell's microtubule organizing centers, or MTOCs, each containing a pair of centrioles, replicate once during late G1 or early S. During prophase, these 2 new centrosomes migrate to opposite poles of the nucleus.
And this sets the whole axis of division.
This migration is the crucial initial event that establishes the ultimate axis and plane of cell division. These centers contain gamma-tubulin, a protein essential for nucleating the growth of the microtubules that will form the spindle.
The actual engagement begins in prometaphase.
Here, the critical event is the attachment of the chromosomes to the spindle via the kinetochore structure we just described. This is not a gentle process.
It's not.
Once attached, the chromosomes move back and forth erratically, almost chaotically, along the spindle fibers at about 0.05 micrometers per second until tension is established.
This chaotic movement eventually leads to the precise order of metaphase.
In metaphase, the chromatid pairs align perfectly along the central metaphase plate. The alignment is geometrically specific and crucial for fidelity.
How so?
A spindle fiber from one pole attaches to the kinetochore of one sister chromatid, and a fiber from the opposite pole attaches to the kinetochore of the other sister chromatid. This biorientation creates tension and ensures accurate separation.
The climax of mitosis is anaphase, the shortest phase where separation finally occurs. It must be absolutely simultaneous across all chromosomes.
Anaphase begins with a simultaneous centromere separation, the physical break that releases the sister chromatids from each other.
What causes that break?
Biochemically, there is evidence suggesting that topoisomerase II, an enzyme crucial for untangling DNA, is involved in unlocking the interlocked, replicated centromere DNA. Mutants lacking this function fail to separate chromosomes.
Once unlocked, what drives the physical movement?
We talk about two subphases here. Anaphase A involves the single chromatid migrating rapidly toward its designated pole, still at about 0.05 micrometers per second. This is the crucial event for accurate genetic partitioning.
And anaphase B.
In some cells, anaphase B also occurs, where the entire spindle structure elongates, pushing the poles further apart, further contributing to cell separation. If an error occurs and both chromatids migrate to the same pole, that is non-disjunction, which leads to serious genetic imbalance.
Finally, we reach telophase, which essentially reverses the process of prophase.
Two new nuclei form. The reconstruction of the nuclear envelope is incredibly elegant. The persisting polymerized lamin B that stayed associated with the membrane fragments,
The ER storage tags,
provides the necessary starting site. Soluble lamins A and C are then dephosphorylated, allowing them to polymerize back onto the lamin B base structure, reassembling the envelope. Pore complexes then assemble within the envelope.
and everything else resets.
Simultaneously, the nucleolus reforms, the mitotic spindle dissolves, and the chromosomes decondense back into diffuse chromatin.
Mitosis has accurately partitioned the nucleus, but the cell isn't truly divided until cytokinesis separates the cytoplasm and the organelles.
In animal cells, this happens via furrowing. You can see it in figure 1216b. It starts as an indentation of the plasma membrane at the cell's former equatorial plate, beginning in late anaphase or telophase.
And the location of that furrow isn't random.
Critically, the plane of furrowing is not arbitrary. Experimental data, particularly from flattening sea urchin eggs, prove that the plane is dictated by the astral centers and the spindle itself, always forming perpendicular to the spindle axis.
And the power for this pinch is the contractile ring.
Yes. Just below the furrow, a transient contractile ring forms rapidly, composed primarily of actin filaments and associated myosin II, which is distinct from the type of myosin found during interphase. This ring contracts like a purse string, pinching the cell in two.
And we know it's an active process.
We know this requires active growth because experimental use of inhibitors like cytokinin B and phalloidin shows that active microfilament growth and contraction are absolutely essential for the fur to progress.
Plant cells have a major structural hurdle, the rigid cell wall. How do they divide?
Plant cytokinesis happens via cell plate formation, which you can see in figure 1216a. Instead of pinching inward, the new wall grows from the center outward.
How does that work?
In early telophase, Golgi-derived vesicles carrying cell wall polysaccharides aggregate at the equatorial plate. These vesicles coalesce, growing outward toward the periphery, forming the new plasma membrane and cell wall.
And that process creates the channels between plant cells, right?
As they fuse, if endoplasmic reticulum cisternae are traversing the area, they prevent the fusion of the cell plate vesicles, resulting in the formation of plasmodesmata, the essential channels connecting adjacent plant cells.
The final puzzle of this section, and perhaps the most mysterious part of mitosis, is the puzzle of chromosome movement. How do they move along the spindle fibers with such precision?
The elegance of anaphase A movement, traveling at that consistent rate of 0.5 micrometers per second, has led to intensive study of the spindle's components, microtubules, associated proteins, and various molecular motors.
There are three major mechanisms proposed. Let's start with the one that feels the most counterintuitive: force through disassembly.
Mechanism 1. Microtubule length change, depolymerization force. This proposes that the force to pull the chromosome to the pole is generated by the disassembly or depolymerization of the spindle fibers themselves.
So it's like the chromosome is riding a railway track that is dissolving beneath it, and the force comes from the track shortening, not an engine pulling it.
That's a great analogy. Microtubules are dynamically unstable, but during anaphase, the uncapped kinetic core end, the plus end, depolymerizes, and the energy released by this structural collapse provides the physical force to pull the chromosome along.
What is the evidence for this?
The photobleaching experiment is key here. Researchers fluorescently label the tubulin in the spindle and use a laser to bleach a spot midway between the chromosome and the pole.
Oh, okay.
During anaphase A, the chromosome moves rapidly toward that bleached spot, while the bleached spot itself remains stationary relative to the pole. If the depolymerization happened at the pole, the spot would move poleward too.
So the action is at the kinetochore.
This implies the depolymerization, and thus the force is generated right at the kinetochore itself. Furthermore, in vitro experiments demonstrate that chromosomes can move along depolymerizing microtubules without requiring ATP.
The next mechanism brings in the traditional molecular engines.
Mechanism 2, microtubule sliding. This is where cross bridges, potentially made of the motor protein dynein, are hypothesized to exist between antiparallel microtubules in the overlap zone. These cross bridges provide mechanochemical energy requiring ATP to make the microtubules slide past one another.
And this sliding is primarily linked to anaphase B, the spindle elongation phase, correct?
Yes, anaphase B clearly requires ATP to push the poles apart, which strongly supports a motor-driven sliding mechanism, in contrast to the potential passive depolymerization force driving anaphase A.
And mechanism 3 puts the motor right on the chromosome itself.
Mechanism 3, MAP motors at the kinetochore. In this model, the microtubules merely provide the rigid railway framework, and the actual motor, a MAP motor like kinesin or cytoplasmic dynein, is located within the kinetochore's collar, as shown in figure 1219.
And it just walks along the microtubule.
This motor actively walks the chromosome along the tubule. Evidence for this comes from the detection of dynein associated with the kinetochore and specific Drosophilic kinesin mutants that affect chromosome movement during meiosis.
Regardless of the mechanism, the force exerted by the spindle is remarkably controlled. Researchers actually measured this force using a tiny glass needle-bending experiment. This seems like a marvel of micromanipulation.
It is a classic piece of biophysics. Researchers carefully calibrate a fine glass needle. Then, during anaphase, they insert the needle into the dividing cell to physically snag a moving chromosome, stopping its migration. They measure the deflection of the needle when the chromosome finally breaks free.
And that gives them the force.
They estimate the maximum force exerted by the spindle, about 7 × 10⁻⁴ dynes.
That's the measured force. Now, what happens when you calculate the theoretical force needed?
When you calculate the theoretical force needed just to overcome the viscous drag of the cytoplasm and move a chromosome, using something like Stokes' law, the result is only about 10⁻⁸ dynes.
Wait, so the measured force is what, 10,000 times greater?
It is 4 orders of magnitude greater than what is actually needed.
Why would evolution build such massive overcapacity? That suggests the system is engineered to deliver 10,000 times the necessary punch.
It strongly implies that the core motor mechanism is incredibly powerful, but that the cell operates with a powerful regulator, a molecular governor, that prevents the chromosomes from being moved too fast or too violently. It's not about raw power, it's about control and redundancy.
That inherent overcapacity and the necessity for precise timing point directly to an incredibly tight system of control. Let's move to the regulation itself. The orderly flow must be governed by sequential dependency.
Like a molecular assembly line, yes. It is based on a genetic program where the sequential expression of key genes dictates the events. While many cellular proteins are expressed constitutively, key structural and enzymatic proteins fluctuate cyclically.
For example,
DNA synthesis enzymes peak in G1S, and tubulin, for the spindle, peaks in G2. In budding yeast alone, roughly 400 genes are directly involved in orchestrating the cycle.
we test this sequential dependency by using inhibitors or mutants and asking, does event B depend on event A having occurred first?
We can use inhibitors to great effect. If we apply colchicine, a microtubule inhibitor, it blocks anaphase and cytokinesis, but all preceding events like DNA replication and prophase chromosome condensation continue unimpeded.
So they're independent.
They are semi-independent of the spindle. Contrast that with DNA synthesis inhibitors, which universally arrest the entire cycle at the G1S boundary. This proves that all subsequent events, G2M cytokinesis, are absolutely dependent on the completion of DNA synthesis.
You can't divide what you haven't copied.
Exactly. You cannot enter mitosis until you confirm you have perfectly doubled your DNA.
The ultimate internal control system governing the two major transitions is the kinase cyclin system, based on the discovery of Maturation Promoting Factor, MPF. This is one of the most important discoveries in modern biology.
The historical context is essential. MPF was discovered by injecting cytoplasm from a mature, dividing frog egg into an immature oocyte. The recipient oocyte immediately resumed the cell cycle and began to divide.
So MPF is the engine that drives the cell into M phase.
It is, and it is a diffusible cytoplasmic factor.
What are the components of this engine and how does it work?
MPF is a protein complex with two key subunits. The core enzyme is a protein kinase, the enzyme that adds phosphate groups to other proteins. Crucially, it has a second subunit that acts as the activator and the cyclin binding site.
And this kinase is always there, but it's usually off.
It's constitutively present, but inert until bound by its activator.
And once activated, the kinase performs specific phosphorylation, triggering the cascade of visible mitotic events.
That's right. Its main phosphorylation targets include, first, histone H1, which leads directly to chromosome condensation. Second, the nuclear lamins, which leads to nuclear envelope breakdown. And third, key proteins involved in microtubule stability,
transitioning them to the unstable state needed for the spindle. The activation mechanism itself is cyclic, which brings in the key regulator, cyclin.
The core insight of the mechanism is that the MPF kinase is constitutively present but inert. Cyclin is the protein that is newly synthesized during interphase S and G2 and accumulates steadily.
And when it hits a certain level,
when it reaches a critical threshold and binds to the kinase at the G2M transition point, the complex is instantaneously activated.
And the signal to end mitosis and re-enter interphase is surprisingly violent?
It is. During mitosis, the cyclin protein is rapidly and aggressively degraded, which instantly inactivates the MPF complex. This loss of activation allows the cell to exit mitosis and re-enter interphase.
And this degradation is essential.
We know this degradation is absolutely essential because if you engineer a truncated cyclin that cannot be degraded, the cell gets permanently arrested in metaphase, unable to finish division. The entire cycle of synthesis, accumulation, activation, and rapid destruction of cyclin acts as the timing clock.
The universality of this system is astounding. We found the exact same mechanism operating in yeast, but with crucial modulators defining the cell size.
Fission yeast confirmed the universality, having homologous genes for the MPF kinase and the cyclin. But they also revealed crucial feedback loops. The Wee gene codes for an inhibitor specifically, a kinase that phosphorylates and inactivates MPF.
And a non-functional Wee mutant?
It can't inactivate MPF, leading to mitosis starting too early, hence the name Wee, resulting in smaller cells.
Conversely, what speeds things up?
Gene 25 codes for an activator, specifically a phosphatase, that removes the phosphate groups added by the Wee gene, thereby activating MPF. If gene 25 is non-functional, cells are unable to activate MPF and arrest permanently in G2.
So it's an exquisite regulatory balance, a push and a pull.
It's a binary switch managed by two opposing enzymes.
And the same system also controls the other major boundary, the G1S transition, or start.
That's the most unifying concept. In budding yeast, a single point in G1 called start dictates whether the cell commits to replication or enters alternative pathways like quiescence or mating.
And it's the same mechanism.
This start point is also governed by a protein kinase, which is homologous to the MPF kinase. It is activated by a specific G1 cyclin. The elegant unity of mechanism, governing both major transitions using homologous kinases and cyclins across diverse organisms, is an unexpected finding that underscores the shared ancestry of all eukaryotic life.
Beyond the internal genetic programming, cell division is obviously dictated by external mitotic inducers in multicellular organisms.
These external factors are crucial for higher eukaryotes, ensuring that cells only divide when they should, to replace a wound or grow in a specific location. One theoretical mechanism is negative feedback via hypothesized endogenous factors called chelons.
What are those?
They're thought to be factors that inhibit mitosis in specific differentiated cells when a tissue reaches sufficient size. Transforming growth factor, or TGF, is a strong candidate, as it inhibits division in some epithelial tumors.
Plants, of course, rely heavily on their own growth factors, the hormones.
Yes, plant hormones are essential for cycle progression. Auxin is needed for the cell to pass through G1. Cytokinin, another hormone, is specifically required for G2 progression.
In animals, we rely on animal growth factors, mitogens, polypeptides, to push cells past start and into the cycle.
These are active at incredibly low concentrations, down to 10 to the minus 10 molar. Take IL-2, interleukin-2, which stimulates T lymphocytes. Its ability to stimulate rapid T-cell division was absolutely invaluable for researchers initially isolating the AIDS virus, as T-cells normally die very quickly in culture.
And another one is EPO.
Right, erythropoietin, or EPO, synthesized with the liver and kidney, which stimulates erythrocyte production in the bone marrow and is now used therapeutically to treat low red cell counts.
How do these growth factors signal the cell to divide when they can't enter the nucleus themselves?
They bind to specific receptors at the cell membrane, which are often connected to tyrosine protein kinases. This receptor factor complex is then internalized, usually into lysosomes for destruction, but the crucial point is that continuous binding at the cell surface is a prerequisite for cell cycle stimulation.
So you need a constant signal.
Even though the complex is internalized, the factor must continuously bind to the surface to keep the signal active, ensuring the cell only commits to division if the external conditions remain favorable.
We've spent most of our time on mitosis, the cycle for somatic cell continuity, the standard operating procedure for every cell in your body. But there is a specialized, high-stakes game for sexual reproduction: meiosis. Let's quickly outline the key ways it breaks the rules of mitosis.
Meiosis shares many mechanical aspects with mitosis G1 and S phases, chromosome condensation, spindle formation, and so on. But the overall result is fundamentally different. It's designed to promote diversity, not identity.
The most fundamental difference is the reduction division.
Right. Mitosis yields 2 cells, each with the diploid number, 46 chromosomes in humans. Meiosis yields 4 cells, each with a haploid number, 23 chromosomes in humans.
Okay, that's difference 1.
Secondly, meiosis occurs only in germ cells, while mitosis occurs in many tissues.
And the third difference is the structure of the division. Why do we get half the DNA?
Mitosis is 1 DNA replication followed by 1 cell division. Meiosis is 1 DNA replication followed by 2 cell divisions, Meiosis I and Meiosis II, with no intervening S phase.
That lack of a second replication is key.
It's what halves the DNA content.
Meiosis also sees unique chromosome behavior pairing up.
Yes. In mitosis, homologous chromosomes behave independently. In meiosis A, they tear up longitudinally and align non-randomly. This pairing facilitates the next difference: recombination, or crossing over, which shuffles genes and creates massive genetic variability.
An event that's rare in mitosis.
Rare and usually deleterious in mitosis. Finally, meiosis is vastly slower weeks in the human testis, months in plants, and incredibly, Prophase I can be arrested for up to 50 years in human oocytes.
Let's detail that complex Prophase I. It has 5 distinct stages, necessary because the chromosomes must find and align with their partners.
It begins with leptonema, where duplicated chromosomes appear as single threads. Then comes zygonema, where homologous chromosomes begin to pair precisely, forming the synaptonomal complex. This complex is essential for physically maintaining pairing and facilitating crossing over.
Then we have pachynyma.
Pairing is complete here, and crucially, recombination breakage and reunion occurs, physically generating new gene linkages. The complex then begins to disappear. This leads to diplonema, where the chromosomes repel but remain attached only at the sites of crossing over the chiasmata.
This is the stage that can last for decades.
In human oocytes, yes. Finally, diakinesis sees the chromosomes condense further and the nuclear envelope disappears.
Moving to metaphase I, the alignment is unique because of the pairing.
The homologous pairs, held together by chiasmata, align at the equatorial plate. The most crucial structural difference compared to mitosis is the kinetochore difference, which you can see in figure 1225. In mitosis, the 2 kinetotores of a single chromosome face opposite poles.
But in meiosis I,
In meiosis I, both kinetotores of a single chromosome face the same pole.
this structural arrangement ensures they migrate together, right?
Exactly. In anaphase I, the homologous pair is separate, with one entire member of the pair consisting of 2 chromatids going to each pole. But the centromeres of the 2 sister chromatids do not separate. They remain stuck together.
And meiosis II is essentially a cleanup division.
It follows telophase I without intervening DNA replication. This division is mitotic-like. The centromeres finally separate, and the sister chromatids migrate to opposite poles, resulting in 4 unique haploid cells.
We started the deep dive talking about continuity and explosive growth, but to maintain the balance of life, we must also address the highly regulated process of cell death.
Growth and replacement are only half the story of development and tissue maintenance. Cell death, or apoptosis, is an active, programmed process essential for regulating mature size and tissue balance. It's not just passive cellular degradation, it's genetically programmed into every organism.
And this is part of normal development.
Absolutely. The nematode C. elegans reaches its adult size through over 200 mitoses, coupled with dozens of specific programmed cell deaths that are necessary for proper tissue structure.
What are the signals that tell a cell to self-destruct?
Apoptotic cells are generally in G0. Signals can be positive, like hormones glucocorticoids stimulating lymphocyte death. Or the signal can be the absence of a necessary growth factor, such as reduced prolactin causing mammary gland tissue destruction post-weaning.
So the cell requires continuous positive signaling to maintain life.
It does, and there's a crucial insight here about certain cancers that relate to preventing death rather than stimulating division.
This is the BCL2 insight.
The mitochondrial protein, BCL2, when overexpressed, actively prevents programmed cell death. Thus, some aggressive lymphomas arise not because of uncontrolled fast division, but because the cells have an inhibited cell death pathway.
They are surviving when they should have died.
Precisely.
Let's look at the stages of apoptosis, which are shown in figure 1226. It's a 3-hour suicide sequence, and the lack of leakage is a defining feature.
Stage 1. A specific endonucleus is activated, breaking down nuclear DNA into fragments, and the chromatin condenses. Stage 2. The cell detaches from its neighbors and shrinks significantly.
Stage 3.
A dramatic ring of intermediate filaments forms around the nucleus. Stage 4. The cell fragments into several small, neat, membrane-bound bodies containing nuclear pieces and intact organelles. Critically, there is no leakage of inflammatory contents into the extracellular medium.
And the cleanup mechanism ensures the body doesn't trigger an immune response.
Stage 5. These fragments are neatly recognized and swiftly endocytosed by neighboring cells or specialized macrophages and recycled in lysosomes. This process ensures the body maintains balance and removes cellular waste without triggering the massive destructive inflammatory response that occurs during passive cell death or necrosis.
Understanding these controls, especially the balance between duplication dictated by cyclin kinases and death prevention controlled by proteins like BCL2, is absolutely key to designing future medical interventions.
Yes, ones that promote specific tumor cell death.
What an incredible journey through the cycles of continuity and controlled destruction. The coordination required is immense, from the precise, factor-driven timing of DNA replication to the engineered, motor-driven separation of chromosomes.
What truly stands out is the elegant and unexpected molecular unity and control. The kinase cyclin system is the master switch, governing not only the G2M transition but also the critical G1S start point, linking cell fate to a core enzymatic process that is conserved from the simplest yeast to the most complex human cell.
And when you look at the mechanics, you realize the incredible regulatory capacity inherent in the system. We saw the spindle motor exerts a maximum force that is 4 orders of magnitude greater than the theoretical force needed to simply move a chromosome.
Right.
That vast difference confirms the existence of a highly regulated governor ensuring flawless execution, regardless of external stresses.
Indeed. We've defined the cycle, detailed the checkpoints, and explored the molecular switches that dictate both life and death. But the ultimate philosophical question that remains, and one for you to continue exploring, is that delicate boundary. We've seen cancer isn't always about faster division, and that development requires scheduled death. Given the intricate, shared kinase mechanisms that dictate duplication and the specific proteins like BCL2 that actively prevent self-destruction, what truly are the molecular boundaries between a cell deciding to duplicate and deciding to commit suicide?
A profound balance.
It's a profound balance that determines existence itself.
A profound question to end on. Thank you for joining us for this deep dive into the complex, perfectly choreographed world of the cell cycle.